In this lecture, I will be introducing the concept of quantitative genetics and start with this very famous photograph of the distribution of heights among a class. I'm not sure what exact university it was from. Women are shown in white, and men are shown in darker colors, and one can see that there's like sort of an approximate normal distribution to heights ranging from 5,0 to all the way to 6,5, and one can see that this normal distribution is basically a composite of two other normal distributions for height for women and then for height for men. So what I want to do is start thinking about traits which are not Mendelian in the classic sense, meaning, you're either this or that, right? You're either white or black. You either are wrinkled or smooth. We're going to talk now about actual traits that are what we call quantitative, meaning they're traits that take on a range of values. It could be height, it could be weight, it could be propensity for risk, all sorts of different quantitative traits, risk of cancer, that would be another one. And so the nature of the genetic basis for quantitative traits, we need to think about it with a different machinery than the machinery that we use for Mendelian traits. So the framework that we have is that we believe that quantitative traits are produced by the contribution of many different genes. So let's suppose that we have 100 genes, and for each gene, having one allele, all right, so you have two alleles, right? But having one of the allele gives you 1 millimeter of height, and having the other gives you 2 millimeters of height. So if you're homozygous for the first allele, you get 1 plus 1 millimeters of height. And if you're homozygous for the other one, you get 2 plus 2 millimeters of height. And if you're heterozygous, you get 1 plus 2, so 3 millimeters of height. So then it's worth thinking about, with that scenario, what would be the genotype of the shortest individual? Or what would be the genotype of the tallest individual? And clearly the genotype of the shortest individual would be the one that is homozygous for all 100 genes for the allele that contributes only 1 millimeter of height. And for the genotype of the tallest, it would be homozygous for the alleles that give 2 millimeters of height. And it's that sort of framework is how we think about quantitative traits and how the sort of normal distribution arises out of it, right? So this is a really important concept in which, if there are many, many different factors that contribute to a trait, and they all have something called identity, they come from the same underlying basis. If you combine all of those different factors that each contribute sort of uniformly, either one way or the other way, you'll end up with a normal distribution. And so you can kind of see this framework here. Here we have a distribution of heights. Here's 62 inches. The mean is about 63 inches here. And then you can see there's a larger distribution. Well, where does that come from? Well, the tails of the distribution, which are, representing lower frequency outcomes, and then the middle is the more abundant frequency, can be sort of approximated by this scenario, which there are, here we have these add 1 unit of height alleles, and then here we have these blue add 2 units of height alleles. If one is a collection and is homozygous and only carries the add 1 unit of height, all right, those individuals would be shorter. And if one has a combination of alleles which all of them add 2 units of height, then those would be individuals that are taller. Now, it's going to be a rare scenario where one has this particular collection of alleles or this particular collection of alleles. But what will be common, there's many different ways in which you can have a middling number, right? You could have 2 of one, and then 1 of the other, and 2 of one, and then 2 of the other, or you could have 1 of one. Anyway, there's many, many, many different ways to arrange the allelic states such that we achieve, middling heights, right? And so that's why the middle frequencies, the middle phenotypes are more abundant than the rare ones, because it sort of requires a bunch of rare events for the either very short individuals or very tall individuals to be identified, to be generated. So this is the general model for how we think of quantitative traits. Now, some traits can have a significant amount of variation, right? So, for example, height. We've been talking about height. Individuals range anywhere from, typically from between 5 and 6 feet. Now, of course, there are, shorter and taller people than that, but typically in that range. But there are also other traits that might have very low variance, right? So that would be, say, for example, core body temperature. And so you can measure that spread with this value called the variance. So here's the distribution in blue. It has a variance is shown with a sigma squared value here, and it's basically how spread out it is. And this is a very, not very spread distribution, 0.25, and that means most individuals are somewhere in that range here. But you can have a distribution that's much more spread out, shown in black here, and this has a variance of 4.0, and there's a much greater number of individuals achieving lower or higher phenotypes. And it's actually the underlying variation is that what we're trying to understand, right? So when we think of height, for example, we'd like to understand what are the genetic basis of the variation, like what explains why some individuals are different than others in terms of height. So what we need to do is we can measure the variation with this term called the variance. Okay, and there's a term called the population variance. So if you took everybody in an entire population and you determined what the mean is, that's what mu is here, all right? And then each individual, you figure out how different they are from the mean. So they take the phenotype of that individual, subtract it from the mean, and then if you square that for each individual in the population and you sum up all of those values and divide by the total number of individuals, you get something called the population variance. And typically it's very difficult to measure the height or any phenotype for every single individual within a population. So oftentimes we measure something called the sample variance instead. Here is the mean of the sample. So this is the mean of the population, but here you have a mean of a sample. And you still have a bunch of individuals in that sample. So you calculate the difference of each of those individuals from the mean of the sample. You square that and sum it, and then you divide. Here you divide by n minus 1 because there's a little bit of a bias that can come out of there, and I'm not going to get into that. But you can divide by n minus 1, which gives you the estimate of the sample variance. And then if you take the square root of that, then that gives you the sample standard deviation shown below. Okay, so let's calculate some sample variances here. The sample variances are what we use to estimate the population level variance. So let's imagine that we're, growing some tomatoes, this big fat beefsteak tomato here, for example, and you grow 8 tomatoes. You grab one tomato from 8 different plants, and you want to see how much variation there is in the tomato weight. Right? So the first tomato is 10 ounces and 12 ounces and 8 ounces, so on and so forth, right? And if you calculate the mean there, what you do is you get 10, right? So the average value is 10. And then from that, for each one of these 8, all right, we figure out how different this individual was from the mean. Now in this particular case, it's equal to the mean, so we do x minus x bar. X bar is the mean. So here that difference is 0, and you square that and you get 0. Right? Here this individual tomato is 12 ounces. The mean was 10 ounces, so 12 minus 10, all right, and then is 2, and then you square that, you get 4, and you just do that, and you sum up all of those things here. Here you have 9, so you get 9 minus 10, which is negative 1, squared is 1. Anyway, you sum it all up, you get 18, and then you divide by n minus 1. So in this particular case, there are 8 tomatoes, so that's 7. So 18 divided by 7, the sample variance is 2.57. Okay, now what is a sample variance here? Well, everybody has the same exact weight, 14, 14, 14, 14, 14, and you can see that, well, in this case, the sample variance is 0, right? Because each of these values is exactly equal to the mean, so there's no variation. So you can see in this case there's no variation, so variance is 0. But here, there's a little bit more variation, and so sample variance here is 2.57. So that measures the spread around the average value. So when we think about traits with respect to plants or animals, what are the factors that contribute to how much variation there is in a population? Some populations might have a lot of variation, and other populations might have very little. And clearly there are two major factors. One of those is genetics, and then the other one is environment. And how does one figure out what the genetic component of variation is? Well, if you control the environment, say for example grow in a greenhouse, then if you measure, say for example, corn, then all the differences that might be identified between the sort of different masses of the corn cobs, if the environment is strictly controlled, as you might do in a greenhouse, then that would be attributable to genetic differences between the different plants. Of course, out in the field, one has a lot of environmental variation. So here's a field of corn, and one can see that this part in front has been flooded, and these corn plants might be under significant stress. So in that field of corn, it's possible that the corn themselves are probably all genetically identical or close to identical because of the way that corn seeds corn is usually sold as a hybrid, and it's a hybrid between two strains of corn that are basically genetically uniform. So the hybrids are genetically uniform. So the differences between these cobs here might be more attributed to environmental variation. All right, so we all know that nature versus nurture, right? Genetics versus environment are the two key things that determine variation in phenotypes. Okay, and so if we break that down more formally, we can kind of think about what are called components of variation, right? So we've been talking about variation. What are the factors that contribute to that variation? Well, the genetic component of variance is actually, you can literally write it as sigma sub g for genetics, and you square it. And one could say, for example, if there were just three genotypes, little a, big A, little a, and big A, big A, if this is their abundance within the population, one can see that if there's no variation in the environment, everybody that has little a genotype has 3 in the phenotype. Everybody that has big A, little a in the genotype has units 5 in the phenotype. And everybody that's big A in the genotype has unit 7 in the phenotype. All of the differences here, all right, between the phenotypes is 100% attributable to genetic variation, right? And so in this particular case, with the way that the distribution is set up, we might have a genotypic variation which is 2, let's say. Okay. Now one could also imagine that if you fix the genotype, so for example, look at only individuals that are little a, or only individuals that are big A, little a, or only individuals that are big A. In the real world, in fact, there will be environmental effects on the phenotype. So the average value for this little a genotype might be 3, but something about the environment may cause some individuals that have that genotype to show a phenotype of 1 or a phenotype of 5. And so this is that environmental variation that is sort of layered on top of the genetic variation. Likewise, individuals big A, little a, there's going to be a spread of phenotypes due to environmental variation. Likewise for big A. And so these two things, the genetic component of variation, which I just showed right here, which is all the differences between these individuals are due to genetics, and the environmental component, so all the differences in these individuals here, because they're genetically identical, little a, is all environmental variation, So those things get layered on top of each other. Here I said that environmental variation was 1. Here I said that genotypic variation was 2. And they actually get added up, and so the variations are additive, right? So what you end up with something called phenotypic variance. So sigma p there for phenotype squared is equal to the genotypic variance plus the environmental variance. So 2 plus 1 equals 3, and that's how we kind of imagine these two factors coming together. So this leads to as geneticists, what we would like to understand is to what degree is a phenotypic variation determined by genetics, right? And that leads us to this concept called the broad sense heritability, all right? We call it big H squared, and we say that that's equal to, and what we have on top is the variation that's due to genetics, the genotypic variation, divided by the sum of the genotypic variation and then the environmental variation, the environmental variance, right? So what this formally is the proportion of phenotypic variance, down below is phenotypic variance, that's due to genetic factors. Now you can think about this for a second. If all the variation is due to genetics, you might have a value of 10 up here, right? You might have a value of 10 down here, and then the amount of variation due to environment might be 0. So in that particular case, you get 10 divided by 10, you get 1. So that would say that all the variation in the phenotype is due to genetics. You might have another case where you have units of 10 variance for genotypic variance, so 10 here and 10 here, but you also have an equal amount of variation that's attributed to the environment, so another 10 units, so it would be 10 divided by 10 plus 10, or 20, so it would be 0.5. Or you can imagine a very extreme case. Well, this might be like 1 or 2 up here, and then this again would be 1 or 2 down here. But a lot of the variance is due to like huge amounts of environmental variation. This could be a very large number here, like 100 or something like that. And then the broad sense heritability would be very small. Okay, so this seems like kind of strange because it seems very theoretical, and to be honest with you, it is fairly theoretical, and it's difficult to determine in populations, right? And the reason is because it requires strict control of the environment or genetic factors, right? So it's easy to do experiments like this, going back with corn, where you can control the environment or you can control the genetics. It's harder to do in for example, human populations, but it can be done. And there are ways in which one can control the environment and genetic factors. And I'll show you briefly how that can be done, but let's talk about how one would estimate broad sense heritability with what are called inbred lines in corn or maize. So this is from the book. In this experiment, the days to pollen shed, all right, so basically how long it takes after a certain time point for pollen to start coming off of the tassels from the top of the corn. Maybe it's from tassel emergence to the release of the pollen. Actually, if it's days out, maybe this is probably actually since it germinated or was planted. Anyway, one can see that one has a bunch of inbred lines, A, B, C, D, and these are so inbred, so they're completely homozygous, and the lines, all the individuals in the line are genetically identical. And one can do this experiment and repeat it across different environments. So in environment 1, line A had 62 days, and B 64 days, so on and so forth. These ones take a little bit longer to mature. But one can see that across the different environments, you see some variation due to environmental variation. So the differences across the environment, 62, 64, 66, or 70, 70, 72, and 74, that's due to environmental variation. But if you take the average of those lines across all those environments, this is due to the genetic differences between these inbred lines. So from an experiment like this, one could estimate the total phenotypic variation, and the book does this, but you could go through the calculation similar to like we did with the beefsteak tomatoes. If one does the full calculation across all these 30 values, so calculate the grand mean, which would be 70, and subtract each of them from the mean and square it, and so on and so forth, you get 14.67 days squared for the total phenotypic variance. The total environmental variance, that's just measured across the values of the environment, the mean, so 68, 70, and 72. So the mean is 70 for that, and then 68 minus 70 squared, and 70 minus 70 squared, so on and so forth, and you get 2.67 days. And then the total genetic variance, which is measured basically the variance across the means, that's 12 days. All right, so you can see that these add up, right? So 12 plus 2.7 equals 14.67, which shows you that the total phenotypic variance is comprised of the total environmental variance and the total genetic variance. And if you calculate broad sense heritability, you get total genetic variance, which is 12.0. Whoops, let me go back. And then that's divided by the sum of 12.0 and 2.67, so you end up 8.82. So, that's a significant amount of the variation is explained by the genetic differences between these inbred lines, and we say that broad sense heritability is 0.82, where it can go from 0 to 1, so that's a pretty large number. Okay, what's important to know, though, is that broad sense heritability depends on the environment. So here, I want you to notice that compared to the previous table, the means for these different environments, they're much more variable, right? All right, 64, 70, and 76, as opposed to 68, 70, and 72. Now, the genetic variation is going to be the same because the means from all of the different lines is going to be the same, but there's just a much more, they didn't maybe do a good enough job of controlling the environment in this experiment. So when one does the grand calculation of the total phenotypic variance, 36 days, and the total environmental variation, you get a much higher variance due to the environment, is 24 days. The genetic variance is the same, 12, right? But if you calculate broad sense heritability, 12 divided by 12 plus 24, or 12 divided by 36, 0.33. And so the important point here is that the genetic component has stayed the same, but the environmental source of variation is much greater. And so it looks like genetics might play less of a role in the variable environment. And that's very, very important to consider when thinking about how one describes the genetic contribution to a trait, because one must also consider the amount of environmental variation. So that's with corn, but one can actually calculate broad sense heritability in humans, and the way that that's done is using identical twins that have been adopted and split up into two different households. Obviously, if identical twins are used for these types of analyses when they're in the same exact environment, one cannot tease apart the environment from the genetics. But if you take identical twins, and oftentimes this happens, not a ton, but it can happen somewhat frequently, if one is adopted by one family and one other of the identical twins is adopted by another family, one has a controlled experiment, basically. And this is how people have estimated broad sense heritability in human populations. And so it's based on this fact. And I won't get into, the book goes into great detail, but I'm not going to worry about that, based on something called a correlation coefficient. So let's just look at, for example, heights of twins. So you might have twin 1 and twin 2, and if their heights are perfectly correlated, so identical twins have the exact same height as each other, right? That means that environment is probably not playing much of a difference when the twins are in different environments, right? If environments were important, then the genetically identical twins might show different heights. But across these different environments, if you have a really perfect correlation, we call it a correlation coefficient of 1.0, and that would suggest, a very strong genetic component. But the correlation can weaken, right? Here it's not perfect, and here there's no correlation at all, and we can go from 1 being perfect correlation to a strong correlation of 0.74 to no correlation. Now it turns out that one can do some math to show that broad sense heritability between the identical twins can be measured with just simply the correlation coefficient. So one takes those identical twins of many, many, many different pairs, and then they would be in different environments, presumably, and calculate the correlation coefficient. And if it's 1, then broader sense heritability is 1. And if it's 0.5, then broader sense heritability is 0.5. Now, what's important here is that we're trying to make an inference not about the genetically identical twins, but we're trying to make a general inference about the genetic contribution of the trait to the broader population. So if your study using these identical twin groups it has to sample the environmental variation as equally as much, right? Because if the environmental variation that the twins experience is different than the environmental variation that exists among, standard people, then the experiment will not estimate the the broad sense heritability correctly. And that could be the case, say, for example, if adoptees from that are identical twins end up tending to be in the same socioeconomic group, that might be a subsample of the broader amount of socioeconomic variation. This would be a problem. So to infer application to a broader population with this type of approach, where you assume that environmental variation experienced by the twins is equal to that in the broader population. And so from these types of studies, scientists over the years have estimated the degree to which broad sense heritability can be found for different traits. So for height, it's 0.88, so that means that about 80 to 90% of variation in height can be explained by genetics. And then you get things like heart rate, 0.49, or extroversion. You can see that these personality attributes might be low. And psychiatric disorders, autism's quite high, but for example, anxiety might be quite low. But I want to, issue a word of caution here that, it's not guaranteed that these experiments always accounted for the environmental component variation in the correct way. But these are our current estimates. Now, that's not the only way one can measure heritability with twins. One can also measure heritability in the correlation between parent and offspring. And there's a different type of heritability that I'm showing here with a little h, not a big H. And I'll describe this more thoroughly shortly. But this is a type of heritability not called broad sense heritability, but called narrow sense heritability. But one can see that, for example, one might find, and could be, that if you had tall parents, they wouldn't necessarily predict tall offspring, and there might not have a really strong correlation between parent and offspring height. And if that's the case, this correlation coefficient would be small, maybe close to 0 even, and that would suggest that heritability, in this case narrow sense heritability, is quite low. But one could also imagine a scenario which height is very, very closely correlated between parents and offspring. And here you might get a very strong correlation coefficient, and you might argue that the heritability of this trait is very high. Or you can imagine some intermediate case. So in addition, you don't always have the opportunity to do these types of twin studies, but one can also measure heritability in these correlations between parents and offspring. And this gives this concept of narrow sense heritability. And narrow sense heritability is another estimate of the genetic component of variance, but it's not derived from twin studies. It's derived from the correlation between parents and offspring. And it's shown with a little h squared. Okay, so for any given trait, one can measure the correlation coefficient. Say for example, offspring in one parent, offspring in the average of the parents, or correlation between half siblings, or first cousins, or identical twins. We've just done that, or full siblings. One can, and then the genetic, the math behind it's a little complicated, so I won't get into this. But if one calculates the correlation coefficient, that correlation coefficient will equal to, in the case of parent and offspring, equal to heritability, narrow sense of heritability divided by 2. So if one sees, for example, the correlation coefficient of 0.5 equal to, so that would be 0.5 equals little h squared divided by 2, that would suggest that heritability is 1, right? And you can see for identical twins, the correlation coefficient, as we said before, is in fact equal to broad sense heritability. Okay, so obviously as the degree of relatedness goes down, one has to divide by a greater and greater factor to recapitulate the correlation coefficient. But this approach can be used. And you don't need to worry about the exact details of these numbers, but I'm showing this table to basically indicate that by measuring correlations between relatives and then doing these sort of additional calculations, one can estimate narrow sense heritability. Okay, and this brings into this concept, not in human populations, but we'll carry it back in a moment. This concept of heritability, not in humans, but for example in breeding systems, right? So breeders are constantly trying to improve their strains or their crops or their breeds. And so I want to walk you through an example here. Let's suppose a breeder is selecting for fat content in pigs. And so this breeder has a population of pigs and starting fat content is 20%. And this breeder wants to breed a pig breed that has higher fat content. And so they select and from that population of 20, and there's variation there, right? They select a group of pigs that have fat content that isn't 20%, but the group has 30%. All right, clearly they're trying to increase fat content. Then one finds out that you breed all the ones that have 30% fat content, and then find that the offspring of the selected pigs, the ones that have 30%, then again have 20% fat content. Would you say that there's high heritability there? No, you would say that there's very low heritability because you started with a population of 20%, you selected a bunch that were 30%, you bred them, and you end up back where you were before at 20%. Alternately, you have a scenario in which you breed these ones for 30% fat content and all of the offspring have 30% fat content. In that case, you would be like, whoa, I have a really good response to selection. My breeding seems to be going well, and it indicates very high heritability. And one can see here, there's a connection now between the parent-offspring correlation, because in the first case, there was very low correlation because 30% fat content pigs were selected and they all ended up being 20% fat content, low correlation. But here, the outcome of the breeding were still 30%, so there's a very strong correlation. And that ties into heritability. Okay, so what this means is that highly heritable traits will have a very strong response to selection. And you can measure this response to selection with something called the breeder's equation. The book does it a little differently, but I like this approach, so we're going to use this. So, big M will be the starting mean, right? Whatever it is, fat content, height, anything. M star is the selected mean. So a selection experiment is done, and the subset of the population is selected, and the mean of that new population has a new value of M star. Then M prime, okay, so this is sort of like how good are you in breeding your pigs or whatever. M prime is the mean of the offspring. And then there's this term, little h squared, which I mentioned a few other times, which we talked about measuring from correlation coefficients between parent and offspring. That's narrow sense heritability. So we want to keep track of four things. Big M star, M prime, and little h squared. Okay, so just to be clear what this means, suppose you have a distribution of phenotypes. You might, take everybody that has a phenotype above this particular threshold, and of that group, you would have a new mean, M star, right, which would be up here. I mean, this isn't the mean of that group. The mean of that group is here because this is your cutoff. So here's the starting mean. The yellow is M star. And then you have something called the selection differential, which is the degree, how strong your selection was, right? M star minus M. And then you also have the mean of the offspring, which is M prime. And you can characterize something called the selection response, which is how different is your selected group from the original mean, M prime minus M. Okay, so let's keep those things in mind. M versus M star, which is the selected, and then the mean of the offspring. So these are all mated, and then this is the new mean. You can see it's shifted to some extent. It hasn't maybe shifted all the way here, right? It started here, you selected here, you ended up here. All right. So there's this thing called the Breeder's equation, which measures this response and puts it all in together. So M prime, which is the mean of the offspring, is equal to M, which is the starting mean, plus, and then we have little h squared here. This is the narrow sense heritability. Just to remind you that the narrow sense heritability can go from 0 to 1 because it's based on those correlation coefficients. And then you multiply that by M star minus M, right? The difference between the selected group and the non-selected group. So let's see what happened, like what's going on here. So if little h squared is 0, that means there's no heritability, right? The genetic component of variation is basically none. Then you'd plug in 0 here, right? And so whatever this is just turns into 0. And then M prime, right, which would just be equal to M, right? So, you know, the next generation mean is exactly equal to the starting mean. That was the case where you selected 30%, but you ended up at 20% again. Mwah, mwah. So one can see that when little h squared is 0, you know, basically there's no change in going from M towards M prime. Now, if little h squared is 1, all right, let's see what happens. We have M, right, and then 1 is right here, and then M prime minus M, that's the difference in the means. That means that the entire difference of the mean, because you're multiplying by 1, gets added to the original mean, and so M prime ends up basically being equal to M star, because M star is just the difference between this here value here is the difference between M star and M. So if little h squared is equal to 1, meaning heritability is 1, then M prime in the progeny mean is equal to M star, the selected mean. And then finally, if little h squared is equal to 0.5, then M prime would end up halfway between M star and M. And you can see now that little h squared basically measures the degree to which one can select for a phenotype, and it's determined, of course, by the contribution of genetics. Okay, so this is a sort of an abstract concept, and I want to kind of narrow it down a little bit. What is this difference between broad sense heritability, which I was calling big H squared, and narrow sense heritability, which I've been calling little h squared? Okay, I'm bringing back the definition of broad sense heritability. Broad sense heritability, big H squared, is a measure of the entire contribution of genetics to phenotypic variation. And that's equal to, so here we have the phenotypic variation that's explained completely by genetics, that's the on the numerator, divided by the phenotypic variation due to genetics plus the phenotypic variation due to the environment. Keeping in mind that if you sum these two things up, you just get all phenotypic variation. So the degree to which genetics contributes to phenotypic variation is broad sense heritability. And it's a theoretical thing that one can measure with twin studies or careful experiments, but it's hard to, those are hard to do. Narrow sense heritability is a different type of measure of heritability. It's a measure of the genetic contribution to a trait that can be measured through the correlation between parent and offspring, right? It shows up in the Breeder's equation because the Breeder's equation provides a relationship between selected parents and the phenotype of the offspring. So, now why are we making this distinction between broad and narrow sense heritability? And to explain that, I need to explain something called the problem of dominance. So, one can have traits that are entirely genetically determined, but are difficult to ascertain through the correlation between parent and offspring. So I'll say this a couple more ways. Dominance hides the genetic contribution to a trait by screwing up parent-offspring correlation. So let's just think of a simple case, right? Here we have these mice, heterozygous big A little a, heterozygous big A little a. Now this particular color of the mice is 100% genetically determined. So broad sense heritability is 1. 100% determined by genetics. However, if one were to actually look at the correlation between parent and offspring, here you have a black mouse, here you have a black mouse, you cross them together, every once in a while you'll see a white mouse. And that's weird, because even though there is a complete genetic determination of the phenotype, clearly the parent-offspring correlation is messed up because both of the parents are black and now you have this white offspring. What's going on here is that dominance is causing a problem because there's an allele here that's hidden, right? So even though it's genetically determined why this individual is white, the allele is hidden in the parents, which leads to a poor correlation between parent and offspring that is due to dominance, and that dominance obscures the genetic component of the trait, because one might see this type of cross and might say, okay, well, it's probably not genetic then, because the parents are not correlated with offspring, even though it clearly is genetic. So, this problem of dominance, right, results in a poor correlation between parent and offspring for certain traits. And that poor correlation for certain traits between parent and offspring impinges on, one, breeding programs, because one is not able to obtain what one might expect from breeding parents of one phenotype. Dominance might yield offspring of an unexpected phenotype. So this can impinge on breeding programs, and it can also impinge on evolution by natural selection. Remember that population of mice that were black that landed on an island with white sand. All right. Now, clearly, when there are these snakes around, it would pay to have the white phenotype. But when these individuals mate, all right, this little A here, a lot of their progeny will be still black, and so this basically sort of hides the little a allele from selection, right? Because it would be nice if these mice were able to experience some benefits of camouflage, and it would be nice if these mice were able to experience some benefits of camouflage, even though only these ones can. And because of that, we have an allele that's beneficial, but it's recessive. And so the only way it can kind of become revealed and increase in frequency in the population if a bunch of these recessive homozygote individuals arise. And as we discussed previously, it takes some time for those individuals to arise in the population. So this dominance impedes natural selection. So another point related to this is that there's this complicated nature of the genetic contribution to trait variation. At the beginning of this lecture, we talked about phenotypic variation that's entirely due to genetics, but it turns out one can break this into different parts, right? One can say that the phenotypic component due to genetics can be due to two components, which is one is called the additive component, and then the other one is called the dominance component. The additive component is the genetic contribution to a trait that is measurable by the correlation between parent and offspring. This is what enables natural selection, and this is what enables efficient breeding, is a correlation between parent and offspring. You have a bunch of parents, you select them, hope the offspring will have a similar phenotype, and the degree to which one is successful is determined by the additive component of variation. There's another component of variation, which is the dominance component, and this is the genetic contribution to a trait that is hidden by dominance and not observed in the correlation between parent and offspring. So an allele may completely determine the phenotype, but the dominance may obscure that. Another thing to keep in mind, and again, when we talk about narrow sense heritability, is we can actually think about it more like this, which is the proportion of variance explained by the additive genetic variance. So again, before we said big H squared was equal to genetic variance divided by genetic plus environmental, or the phenotypic variance. Here, little h squared is smaller than big H squared because up above, instead of the entire genetic component plus the dominance component, we just have the additive component, which is the part that is due to the correlation between the parent and offspring. So The proportion of variance explained by additive genetic variance is known as narrow sense heritability, which is why it's called narrow sense, because it's a narrower form of heritability. Another fact that's complicating the genetic component of variation is something called an interaction. All right. So before we said, oh, it's just a sum of genetics and environment, but there's a complicated thing because sometimes the genotype can interact with the environment, and that term is you can't separate it out. And what do I mean by an interaction between a genotype and an environment? It means that certain genotypes might respond to certain environments in unique ways that other genotypes don't, right? It's not like everybody uniformly is affected by the environment the same way, right? There are some people that smoke cigarettes and don't get lung cancer because they may have a certain genotype that makes them less likely to get lung cancer. But the same environment of smoke might cause someone else to get lung cancer. So an individual might have genes that make them only, for example, very tall in certain environments, such as one with a certain diet. And so one needs to consider those types of interactions as part of this contribution to phenotypic variation.